Hello everyone. In this video we are going to discuss the animal diversity. So let's start. So this is an introduction slide about animal diversity and how can we define animals. So animals are multicellular heterotrophic eukaryotes with tissues that develop from embryonic layers. So we are going to dig deeper in this definition in the next slides by showing more characteristics of animals and how they have developed from the embryonic layers. So as I said in this slide, again we are continuing with the characteristics of animals. So they are multicellular eukaryotes. There are heterotrophs that ingest their food, and as you can see now we are starting to create some differences from other groups of eukaryotes, for example fungi. So fungi are also heterotrophs, but they absorb their food. Animals, they lack cell walls, and again this is a different from fungi, which has a chitin cell walls, or from plants which have cellulosic cell walls. Then the next characteristic is that animal tissues develop from embryonic germ layers, and as I have said, we will focus a little bit on this in this video, the development of animal tissues from the embryonic germ layers, because this is a characteristic of animals, and it also helps us to categorize animals into different groups. Among these tissues we have two that are unique for animals, and they are among the defining characteristics of animals, which are the nervous and the muscle tissues. So please keep in mind that the movement in animals require the presence of muscle tissues and the nervous tissues to coordinate this movement. So moving on to reproduction and development, and the reason behind showing reproduction and development, as I said, is number one, understanding the reproduction and development, and number two, because this will later on, as you will see, will help us in categorizing animals into the different groups based on how they develop from their embryonic tissues. So most animals reproduce sexually, and they have the dominant diploid stage. So in their life cycle, the dominant stage will be the diploid, or two copies of the chromosomes. And what will happen is that after fertilization we will reach the 8-cell stage. So from the fertilized egg we will have now a zygote. This zygote will start as 8-cell stage. Then we will go through cleavage. So what does cleavage mean? It means that the zygote will undergo a rapid cell division. So this 8 cell will become 16, then 32 cells, and so on. So by going through cycles of cell division, rapid cell division, we will reach what is known as blastula. So this continues till we reach the blastula, which is like a hollow ball of cells. So if we take a cross section of the blastula, we will see it like that, and we'll see the hollow ball of cells. And this blastula is composed of one layer of cells. Then the blastula will undergo gastrulation. So what is happening here, as you can see in this figure, is the invagination of the blastula. So it will bend inwards like that, and now going from one layer blastula, we are now having a multi-layer gastrula, because later on we will call the resulting structure, we will call it gastrula. So the difference is that the gastrula is composed of multi-layers of cells. As we will see later on, these are called the germline cells or germline layers, and this is important as I said, in characterizing the different groups of animals, which will be shown in the next slides. So animals can be characterized by body plans. So what does a body plan mean? It means a set of morphological and developmental traits. So gastrulation, which we have shown in the previous slide, is conserved among the diverse groups of animals, but the distribution of the organs and the tissues and the development of the mouth and the anus, which one comes first, all of these characteristics differs among the diverse groups of animals. That's why we use these morphological and developmental traits in order to characterize the body plan. So the first feature is the symmetry. So animals can be classified as having bilateral or radial symmetry. When we move to the second feature, the tissues, some are characterized as being diploplastic, while others are triploplastic, and we will cover this feature and the rest of the features in the next slides. So I'm just here showing like the topics that we will cover in the next slides. Then the third feature in the body plan is called body cavities. So this will allow us to classify animals into a coelomate, pseudocoelomate, and then the true coelomate. So body cavity is a coelom, and those having a true coelom, we'll call them coelomate. Those without a coelom will be called a coelomate. Then we have something like in between, we call it like pseudocoelomate. Then in terms of development, we have what is known as protostome versus what is known as deuterostome. So as I said, these four features combined, they make what is known as body plan, and this set of features allow us to characterize animals and set them into the different groups. So starting with the first feature, the symmetry. Some animals have asymmetrical body, for example the sponges. So if you imagine a plane that divides this sponge, so the two halves will not be identical, so all the possible planes will not result in a symmetrical body plan. Some animals have radial symmetry, so there is no front, no back, no left, no right, and here is one of the examples. So there are indefinite number of planes. Each one of them can divide the animal into two equal or identical parts, or halves I mean. So these halves are exactly identical, so there is no differences, there is no front or back, right or left, because of the identity of the two halves resulting from the radial symmetry. These radial animals are often sessile or planktonic, so they drift or swim weakly with the current of water. So usually you will find these symmetrical radial symmetrical animals in the sea, for example the hydra and the different types of aquatic animals. Then moving on symmetry. So we started with animals that they don't have symmetry. Some others, they have radial symmetry, and the third group is what we call as bilateral symmetry. So these can be characterized as animals showing two-sided symmetry, something like that. And this bilateral symmetry will generate a dorsal side. So this is the top side, and it will generate the ventral or the bottom side. It will also generate right and left sides and anterior and posterior ends. So here we will have the anterior end, and here we will have the posterior end, and we will have cephalization. So cephalization means the development of a head. So it is the place where the neurons will be collected there. Some animals, they have sensory organs as well, and usually the bilateral animals, they use this head, as we said, for sensing their environment, and this is one different from the radial animals. So radial animals, they feel the environment from all the sides, so they sense the environment from every direction because they don't have an anterior end or a cephalization of the head, while the two-sided animals, the presence of the nerves at the anterior or the head part and the development of the head requires that, or makes it common that these animals use these organs to sense their environment. So if we have a stimulus from the back, we tend to turn around to feel it and to interact with it, and this is the way we interact with our environment. Then the third characteristic in terms of the body plan is the tissues. So tissues are a group of cells that have a common structure or function or both. Some animals, like the sponges, they lack true tissues, but the majority of other animals, they undergo layering and development of different tissues during the embryonic development stage, and these layers are called the germ layers. So as we said before, this is a process that we'll find in most of the animals, and this will generate the different tissues of the animals. We have 2 layers at least. So some animals, they have 3 layers of these germ layers, but the others, they have only 2. So we will start with the 2 layers that you will find in the animals with germ layers. So number 1, the ectoderm. So the ectoderm is the outer layer that covers the embryo surface. So remember when we said we will have a process called gastrulation. So gastrulation is the invagination of the blastula. So this invagination created a layer outside. This is called the ectoderm. So the ectoderm, this layer here, is the outer layer, or the germ outer layer that covers the embryo surface. And then we have what is known as endoderm, which is shown here in yellow. So the endoderm would be the internal layer that, or the innermost layer of the germ layers. So this endoderm covers the archenteron. So archenteron is the primitive gut system or digestive system of the animal. So this is known as archenteron, the digestive tube that is starting to develop. So the layer that will directly cover this archenteron is the endoderm. This is the innermost germ layer. Then we have the outermost germ layer, which is called the ectoderm. So if an animal has these 2 layers, the ectoderm and the endoderm, we will call this animal as being a diploplastic. So diploplastic animals, they have these 2 layers, and you know that diplo means 2. Then triploplastic animals, they have an additional layer called the mesoderm. So this characteristic, we'll find it in those animals which show bilateral symmetry.